Start?
We can start.
All right. Das, you set?
Yeah, I'm sorry. Yeah.
All right, let's go ahead. So, hello and good evening, everyone. Welcome back to CCMB Biologue, the sixth in our series. For those of you who are new to this platform, Biologue is a virtual webinar organized by our students, where they network with leading scientists around the world and invite them to talk about their work. These talks are hosted live and also streamed on our YouTube channel, and we will take questions from YouTube channel participants as well. The typical format is about 45 minutes for the talk, followed by a question and answer session and interaction with the speaker for a while. Today, we have a very special speaker, my colleague from InStem Banglore, Dr. Dasaradhi Palakodeti, or Das as he is called. Das obtained his bachelor's here at the Osmania University and his master's at the Madurai Kamraj University. His PhD was at ICGB, investigating cysteine proteases in the malarial parasite Plasmodium using RNA techniques. During his postdoctoral work at the University of Connecticut, he shifted his model system to study the role of small RNAs and RNA binding proteins involved in stem cell function and regeneration in planarians, where he also had a lot of NGS and bioinformatics experience working on the planaria genome. At InStem, he has continued in this field in the past decade or so, and his lab focuses on RNA biology using planaria as a model system to understand stem cell function and regeneration. And today he's going to talk to us about some of his exciting work on tRNA-derived small RNA fragments in stem cell function. Thank you so much, Das, for accepting our invite, and we really look forward to hearing about your work. Screen is yours.
Thank you very much. And let me put it on a slideshow. So first I want to thank Rakesh and Surabhi and all the student and the postdoc community for inviting me here. And I also learned quite a bit about the CCMB Biologue. It's a very interesting initiative that was started by CCMB. So today I'll be talking about this interesting class of small RNAs that are derived from tRNAs and their function in stem cells. So before I go into the talk, I would like to take small amount of time to discuss what's the focus of my lab. So my lab is interested in understanding the initial events that are critical for stem cell state transitions. So what do I mean by stem cell state transitions? When you take a stem cell and give it a signal, the stem cells undergo differentiation. So the first thing they do is they change their states and further they differentiate down, right? So as the stem cells are undergoing the state changes, what do they have to do? The first thing is change the protein repertoire, which it could be a structural protein, it could be a functional protein that is essential to maintain stem as a stem state, right? So that allows these stem cells to move out from the stem state. So the way the protein repertoire can be changed is by either degrading the existing proteins, or the other way of doing it is by regulating the translation or protein synthesis. Both of them go hand in hand because if you are degrading the protein and at the same time you are making new proteins, it becomes counterproductive to the cell. One way of doing that is you also regulate protein synthesis, and that is what my lab looks at, right? So what do we look as a model systems? We look at both in the context of regeneration. We use this beautiful model called planaria, and I will talk about it in a while. And we also use embryonic stem cells as a means to understand how the stem cell differentiation is regulated. So as I said, we look at translational regulation, and this is a very, what I call it as a snapshot of protein synthesis. This also brings out the feature that the protein synthesis can be regulated at 3 different stages. The first stage is translation initiation, where ribosomes are loaded at the start codon, right? And the second stage is an elongation, where ribosome runs over the mRNA. The third stage is at the translational termination stage, where ribosomes dissociate into the subunits and reassociate back at the initiation. So you can regulate translation at these 3 stages, right? So just to increase the complexity of this process, if you look at this diagram, you can see that there are cis elements and there are trans elements. Trans elements are these RNA binding proteins that actually bind to transcripts and regulate, and cis elements are the secondary structures and G quadruplex structures and also sequence specific structures that are present, which actually regulates how translation is happening, right? So in last, say, 5 to 10 years, my lab has looked at several different kinds of RNA binding proteins that regulate how translation initiation happens in the context of both regeneration and stem cells. And we also looked at some of the cis element factors, such as the length of a poly A tail. So the whole idea is if you change the length of a poly A tail, you change the stability of the mRNA and thus change how the protein synthesis is happening. And we also looked at the sequence-specific elements to which the small RNAs bind, which we all know as microRNAs, and how this microRNAs binding to 3' UTRs regulate the process of regeneration. So today, what I will be going to talk about is this a special class of small RNAs called tRNA-derived small RNAs. How do they regulate translation? And by regulating translation, how do they bring about stem cell state transitions? That is what I'm going to talk about. So that's the focus of the talk. The focus of the talk is look at tRNA-derived small RNAs and their function in embryonic stem cells. And how is this biogenesis regulated? We are talking about a fundamental molecule like tRNA, which carries amino acid, which is critical for protein synthesis, right? Now we are saying that this tRNA is being broken down. So I will give you some aspects of biogenesis here, not the complete mechanism. And here, I also want to show that this is not specific to mammals. This is a very conserved phenomena. And we have shown this in the context of planaria and regeneration. So my talk is divided into 3 parts here. So going back to what is tRNAs? People have shown, the recent studies have shown that the tRNAs can be broken down into multiple pieces. They can be derived into 18 to 22 nucleotide species, and that can come both from the 5' arm and from the 3' arm. And you also have a bigger fragments that are around 25 to 34 nucleotide in length that come from the 5' arm. So today, most of the focus of my talk is on this 25 to 34 nucleotide species that is derived from the 5' arm. And it has been shown that this piece of tsRNAs are mostly upregulated during stress, and they have shown to be having an anti-cancer activity. They were also shown to have a neuroprotective role. And nowadays, these tsRNAs, which are derived from the 5' arm, are being shown as a biomarker. And there is also a transcriptional sequencing role that is associated to that. So we are now. Sorry, is this a question for me? So what we are today showing is the role of this 25 to 34 nucleotide species in the context of embryonic stem cells. So what we have done is this: we have taken ESCs, and ESCs are maintained on leaf, right? And if you differentiate them by putting retinoic acid, they differentiate into neural progenitors. And you take the ESC and treat them in Win3A, they go into a ground state. So we took these 3 cell types. One is ESC, Win3A treated, and RA treated, where the ESCs are differentiating within 48 hours of differentiation by treating with RA. And we have done the small RNA profiling, and we are interested to look at microRNAs and other small RNAs, but we interestingly found that there is a peak that comes around 31 to 34 nucleotide species, right? First, we thought they are piRNAs, and we took them and we just mapped them to a different non-coding RNA species. And what we interestingly found is most of these 31 to 34 nucleotide species are started mapping to the tRNA population. And they steadily increase as the cells undergo differentiation. So the first thing is maybe it's a degradation product, right? But what we interestingly found is if you take these tsRNAs and map them to the tRNA, we find that most of this tsRNA map to the 5' end, and their mapping ends right before the anticodon arm, suggesting that there could be a function associated to this tsRNA, and we have validated them both by Northerns as well as qPCRs. So now the big question is, do they have a function in ESC? R is just a consequence as the ESCs are differentiating, right? So in order to address that, we have used antisense oligos. What we have done here is this: we took ESCs, we treated the ESCs with antisense oligos, and we put them in retinoic acid and allowed them to differentiate. ESCs are typically measured by their alkaline phosphatase activity. As the ESCs differentiate, the alkaline phosphatase activities go down. So if the antisense oligos block tsRNAs, what you should see is the ESC should show an increased levels of alkaline phosphatase in spite of treating with retinoic acid. And that is what exactly we found. We found that when you take an ESC and treat them with retinoic acid, and put them with antisense oligos, you actually see an increase in the alkaline phosphatase rather than the decrease, suggesting that these tRNAs might have a role in the ESC function or in the context of differentiation. The contrary experiment to that is this, right? Now you take ESCs, put them in leaf, so they are now stem state. Now you treat them with a mimic, that means you're overexpressing the tRNA. So what the tRNA should do, if they're essential for differentiation, is push the ESCs towards the differentiation mode. That means you should see a reduction in the alkaline phosphatase activity, though you put them in leaf. And that is what we have seen. What we have seen is when you take an ESCs and treat them with mimics, what we see is a decrease in the alkaline phosphatase activity. So what these two experiments suggest to us is the tRNAs that we found in ESCs indeed have a functional role in controlling how ESCs are undergoing state transitions, or rather say how they undergo differentiation. So the story so far is we identified this 30 to 34 nucleotide small RNA species. They are derived from the 5' arm, and they are enriched as the ESCs are differentiating in retinoic acid treated condition. And what I have not shown here is we have shown that these tRNAs are strongly associated with polysomes, right? Suggesting that their role might be by regulating the ribosomes. And we also have shown, which I haven't shown the data here, is they are general suppressors of translation. And by using antisense oligos and mimic, we have shown that they have a function in the context of how the stem cells differentiate. So one of the big questions is, how are these tRNAs regulated, right? And what is the functions associated to them, right? So the two ways to look at it is to just look at the interactome of the tRNA. So what we are saying is tRNAs have a function in driving the stem cell state transitions. How do you know the function of it? Is by just looking at the transcriptome of it or the interactome of it. One interactome that we are interested to look at is, does this tRNAs interact with specific transcripts, is one way to look at it. The other way to look at it is, does tRNAs interact with any specific proteins? So we have done both here. So for this study, this is what we have done. We took ESCs in leaf, we took ESCs in retinoic acid, which was 48 hours post-treatment. We made a cell extract, we biotilated the tRNA, which is a mimic here, and we have put it in the extract, and we have pulled down with streptavidin, and we have done the transcriptome sequencing. What we have found is, both in the ESC and the RA treated condition, the tRNAs are interacting with a specific sets of transcripts, right? In a ESCs, you have a specific sets of transcripts it's interacting with, and in a RA treated condition, it also interacts with a different set of transcripts. So now when you go back and look at the nature of this transcript, this is what we found. In a leaf treated condition, which is ESC condition, the tRNAs are interacting more with a differentiation responsive genes. They are not interacting with the pluripotency genes, suggesting that the tRNAs might be interacting with the transcripts that are essential for differentiation and blocking them, and thereby maintaining the stem state. This is one hypothesis. And when you go back and look in a RA-treated condition, what we found is, we found that the tRNAs in a RA condition are now interacting with a specific pluripotency associated genes. And what we also found is they are still interacting with differentiation responsive genes. When we went back and looked at what is this differentiation responsive genes, what we found is the differentiation responsive genes are the genes that are critical for mesodermal fate and endodermal fate. What is happening when you treat with retinoic acid is the ESCs undergo neuroectodermal differentiation. So what these tRNAs are interacting with are, they're interacting with the transcripts that are critical for mesodermal fate and endodermal fate. So what is this telling us is, the tRNAs are not just essential for stem cell state transition. The tRNAs that are enriched in retinoic acid condition are also important for linear specification by interacting with specific sets of transcripts. So now one of the $1 million question is, if tRNAs are general suppressors of translation, how is the specificity coming? One way to look at the specificity is there a complementarity? We have used extensive bioinformatics, and we found that there is no sequence complementarity between the tRNAs and the transcript. fragments. At least we couldn't find it. The other way the specificity can come is by interacting with a specific set of RNA binding proteins. So what we did next is we did the same experiment now, and instead of doing a transcriptome sequencing, we have done a proteome sequencing. And what we interestingly found is in a retinoic acid treated condition, the tRNAs are interacting with a specific set of RNA binding proteins, and one of the top candidates where tRNAs are binding in the presence of retinoic acid is this molecule called IGF-2BP1. And this is what I'll be going to talk about. So what is this IGF-2BP1? Which is interacting with tRNAs in a retinoic acid treated condition post 48 hours. So IGF-2BP1 is insulin-like growth factor 2 mRNA binding protein. It's actually an RNA binding protein. It is highly enriched in the cancer cells. And if you block IGF-2BP1 in cancer cells, you actually block the proliferation. So what is the mechanism that we know about IGF-2BP1 is very interesting. IGF-2BP1, along with other RNA binding proteins, bind to MYC mRNA. And the binding of IGF-2BP1 to MYC mRNA is essential for stabilizing the MYC mRNA. And this binding of IGF-2BP1 to MYC mRNA loads the MYC mRNA into the ribosome, right? So now, and thus it translates the c-MYC mRNA and the protein is made. So now we have two working hypotheses is this, right? In a lift condition or in ESC state, IGF-2BP1 stabilizes c-MYC, loads it onto the ribosome, translation happens, and c-MYC is formed. What is the c-MYC? c-MYC is a cell cycle regulator, and it is essential for the proliferation. So if you block, you stop proliferation, and you push ESCs towards the differentiation. In a retinoic acid condition, this is our hypothesis. What we think happening is the tRNAs goes and binds to IGF-2BP1 and dislodges IGF-2BP1 from the c-MYC mRNA and thus stop the process of translation. So how do we show this? To show that, we need to prove three conditions here, right? One condition is to show that IGF-2BP1 selectively interacts with tRNA in a retinoic acid condition. That's the first thing we have to prove. The second thing we have to prove is this. In a RA-treated condition, the c-MYC mRNA should dislodge from IGF-2BP1 and go to a non-translating complex. That is how you block the protein synthesis, right? And the third experiment is that the c-MYC RNA should have a lesser association with the translation complex. Or in other ways, the third experiment is that the c-MYC association with the ribosomes should go down in a retinoic acid treated condition. If we show these three, it proves that indeed tRNAs interact with IGF-2BP1 and blocks the process of translation. So that is what we have done. We took ESC, we took RA, we have done pull down with IGF-2BP1. One pool, we have isolated small RNAs and looked for the association with tRNA. In other pool, we looked for its association with c-MYC mRNA. So what we found is this. We found that the IGF-2BP1 heavily interacts with tRNA in a RA-treated condition. So this proves that indeed in RA-treated condition, IGF-2BP1 is interacting with the c-MYC mRNA. Sorry, is interacting with the tRNAs. The second experiment is this. The second experiment is in a RA-treated condition, the association of c-MYC mRNA with IGF-2BP1 should go down. And that is what exactly we have seen. What we have seen is in a RA-treated condition, you see a reduction in the association of c-MYC mRNA with IGF-2BP1. So now the third experiment is this. The third experiment is in a RA-treated condition, the c-MYC mRNA should dislodge from the ribosomes and go into a non-translating complex. To show the third experiment, what we have done is we took ESCs, we took retinoic acid, we did polysome profiling, we isolated non-translating complex and translating complex and asked between ESC and RA-treated condition, how is c-MYC mRNA moving, right? And what we interestingly found is this. We found that in a RA-treated condition, the c-MYC mRNA actually goes into a non-translating complex rather than the translating complex, which is evident from the graph that you see. You see that ATS and polysome, there's a reduction in association with the ribosomes for c-MYC mRNA, and the c-MYC mRNA is now enriching in a non-translating complex in the case of the RA-treated conditions. So these three experiments prove our hypothesis that in a RA-treated condition, tRNAs are interacting with IGF-2BP1, dislodging IGF-2BP1, and blocking the translation of c-MYC mRNA. How do you prove? What is the nail in the coffin experiment is? Now you take this and now you treat with the antisense oligos. If we treat with the antisense oligos, we need to revert back this process, right? And that is what we have done. We took ESC, treated with retinoic acid. We took two pools. One pool of RA-treated condition, we added antisense oligo. In another, we just continued with the experiment. We did polysome profiling, both from RA-treated plus ASO, and asked the question, what is happening to this c-MYC mRNA there? What we found is this: when you take the retinoic acid, antisense oligo treated, and normalize it by RA, what we found is in an antisense oligo treated condition, the c-MYC mRNA is moving back into the translating complex, which is evident from the graph here, where you see an increase of c-MYC mRNA with ribosomes, both in the ATS as well as in the polysomes, and its association with the non-translating complex has decreased. These experiments together show that the tRNAs indeed regulate the c-MYC mRNA translation by its binding to the RNA binding protein, which is IGF-2BP1, critical for the stability of c-MYC mRNA and subsequently for its translation. So this is the first part of the study. And what does this study interestingly show is this: if you take an ESC and allow the ESCs to differentiate, as the ESCs undergo state transition, what we typically found is we found a decrease in the stemness genes, translation of the stemness genes, and we see an increase in the tRNA population, suggesting that these tRNAs indeed bind to the translation machinery of stemness genes, block them. As a result, they push ESCs towards differentiation, and we subsequently see an increase in the translation of a differentiation marker, suggesting that tRNAs indeed are the molecules that are critical for the stem cell state transitions, right? So what next? Now I'm talking about a scenario where you have a tRNA, that is being broken down into a tRNA, and this tRNA is regulating the stem cell state transition. But when you do this, you should understand that you cannot deplete the tRNA, because if you deplete the tRNA, it blocks overall translation. That means there should be a balance in terms of how much tRNA is being converted into tRNA. And we typically find that only 2 to 3% of the tRNA is actually being controlled into a tRNA. So now the question is, how is this balance being happened? Not only that, what we also found is this. Typically in the literature, it has been shown that the tRNA is processed by this endonuclease called angiogenin. The angiogenin is an endonuclease that cuts the tRNA to generate a 5' tRNA. And when angiogenin cuts the tRNA to generate 5' tRNA, it keeps this modification, which is called 2', 3' cyclic phosphate modification. In order to sequence these fragments, you need to use a T4 polynucleotide kinase in order to remove that cyclic phosphate group. Then only you can sequence it, right? The other way of sequencing is you just take tRNA and do a conventional sequencing. But when we initially did the sequencing, we didn't treat with T4 nucleotide kinase, right? So now what we have done is we have done both kinds of sequencing. What interestingly came out from the study is this. We found that the tRNAs that go up during stem cell state transitions are actually angiogenin independent tRNAs. That means those tRNAs are not being processed by angiogenin. And we also found a second pool of tRNAs that are angiogenin dependent. And they actually go down during differentiation and come up at the later stages. So now, just to add further complexity to the biogenesis part, now you have a tRNA molecules, right? And we are saying that one tRNA molecule can be processed by angiogenin and generate a 5' tRNA fragment. And there is another tRNA molecule that is being processed by an endoribonuclease, which we don't know now, and is also generating a 5' tRNA. So now, and both of them exist in a stem condition. So now, just to add to the confusion of regulation of biogenesis, how is these two enzymes deciding what tRNAs has to be cut and how do we generate the 5' tRNAs, right? The answer for that lies in the modifications of the tRNA. We know that the tRNAs are a heavily modified molecule inside a cell, RNA molecule inside a cell. If you change the modifications, you can actually bring about a differential processing of tRNA into tsRNA. And it has been beautifully shown by Banglore et al. In 2014 that an angiogenin, which is an endoribonuclease, typically cuts tRNA to generate tsRNA, and an enzyme, which is a methyltransferase, if it adds a methyl group to the cytosine just before the anticodon, it actually blocks the processing of angiogenin. As a result, the tRNA is not processed. So just by regulating how the methylation is happening, you can change how the tRNA is being processed into tsRNA. So this regulation of methylation can happen at two levels. One is you just change the expression of the enzymes that are essential for methylation, demethylation, and thiolations, right? And bring about changes in the modifications. The other way to regulate is change the metabolites that are critical for the methylation and demethylation. That also can bring about changes in how tRNAs are being modified and generate the tsRNAs. So we first went and looked at the enzyme levels that are critical both for thiolation, demethylation, and methylation. In a nutshell, we haven't found any difference in the enzyme expression patterns between the LIF condition and RA 48-hour condition, which is shown by the arrow marks here. These are the enzymes that are essential for thiolation. We didn't see any change in their expression. We also looked for demethylases and methylases. Again, you can see that red arrow marks there. We haven't seen any difference in the expression of enzymes. So that brings us to the third point, is the availability of the metabolites. For methylation, we know that the cofactor that is needed is a SAM, and the SAM is generated by a methionine. So the presence of methionine makes SAM, and SAM is a cofactor. This is a methyl donor for methyl transferases. And other way of regulating methylation is by changing demethyl transferase activity. And one of the key cofactors that are essential for demethyl transferases, which belongs to this Jumanji family of demethylases, is alpha ketoglutarate. So just by changing the metabolite levels inside a cell, you can change how the tRNAs is being processed. And this is not the first thing that we are looking at. It has been previously published that if you tweak the methionine levels in a culture medium, you can actually push the pluripotent stem cells to undergo differentiation. But in that study, they have talked a lot about epigenetic changes, but they haven't really looked at how tRNA modifications are changing. And recently, Sunil Lakshman from InStem has published this beautiful paper in eLife, where he has shown that the tRNA can be a sensor in terms of how the metabolic regulation can happen, right? So taking all this study into place, the hypothesis is change the metabolites and change how the tRNAs are being processed. In order to find that out, what we have done is we took ESCs and we have depleted ESCs, we depleted methionine in the culture medium. And when you come to 0.25 millimolar state of methionine in the culture medium, you start differentiating the ESCs, which is measured by the alkaline phosphatase staining and also by the colony size here. So that's the concentration now we have used for our study. So what we have done is we took a complete medium where ESCs are growing. We depleted methionine to 0.25 millimolar concentrations, and we have done small RNA sequencing. And very interestingly, we found 3 populations of tsRNAs. One population that is upregulated when you deplete methionine, which makes sense because now when you deplete methionine, you don't have methylation, the tRNAs are being processed, and you see increase in tRNAs. What is interesting is the tRNAs that don't undergo any change. And what is more interesting is when you actually deplete methionine, instead of tRNAs going up, we also saw a pool of tRNAs that actually go down. When we took the upregulated lot and downregulated lot and look for base residue preferences, what we found is this. We found that the tRNAs have different base residues. For instance, you can see here at the 11th position, you have cytosine here, and at the 16th position, you have thymine, and at 11th position, you have thymine in uridine here in case of tRNA, and at 16th position, you have uridine there, right? So suggests that these tRNAs that are being, the tRNAs that are being upregulated and downregulated are coming from two different parent tRNA molecules. So if you now take the upregulated, downregulated tRNAs in a methionine depleted condition, and these are the top candidates. What caught our attention is that if you look at this is a TS arginine TCG, and you in a downregulated you also have TS arginine CCG. That means these are coming from two parent tRNAs which has different anticodons but code for arginine. In one case, it's upregulated, in other case, it's downregulated. When you just go back and look at this tRNAs, what you found is this. In case of TS arginine TCG, where the tRNA is upregulated, you have a cytosine just before the anticodon arm. And that cytosine can be methylated. So now you know that if you deplete methionine, that residue is not getting methylated. As a result, the tRNAs coming from that tRNA is going up. Now, the other one is the TS arginine CCG. There is no cytosine, there is a uridine there. So now this tRNA is not actually getting processed. As a result, it's getting downregulated. That means the parent tRNA is not being processed by the enzyme in order to make the tRNAs. The other interesting example is TS alanine AGC and TS alanine CGC. If you see that, if you look at the ones that are downregulated, there are multiple cytosines that can be methylated, right? This is a very typical case that tRNA is being downregulated in spite of having cytosines that can be methylated at multiple positions. This suggests that in one case, there could be a hypomethylation, in other case, there could be a hypermethylation. And changes in these methylation strategies, depending on where the methylation is happening on the residue, you can have either upregulation or downregulation of the tRNA. So the current working hypothesis is this. If you change the metabolic levels, you can change the way the modifications are happening, and where the modification is happening determines whether the tRNA population goes up or the tRNA population goes down. And these tRNAs are coming from different parent tRNAs. So this suggests that regulating a metabolic levels inside a cell will change the tRNA to make tRNAs, and these tRNAs are critical for driving the stem cell state transitions. So this is where we are, and we are doing extensive mass spec analysis on the tRNAs to identify where these modifications are exactly happening. And if we tweak those modifications, how it is going to affect the stem cell state transition is what we are currently working with. So this brings me to the last part of my talk, which I will take another maybe 5 to 10 minutes and wrap it up, is this tRNA specific to ESCs or mammals, right? Because we are working with mammalian ESCs. Are they conserved across metazoans? So in order to address this, the other question is this tRNA essential for stem cell state transitions in also the other metazoans, right? So in order to address that, we used a model system called planaria. Planaria is a flat form. They grow in fresh waters. And why we choose this model system? Because you can take this planaria and you cut it into small pieces, and each piece can regenerate back into a complete animal. And it has been shown that this planaria carries this bag of stem cells which are pluripotent in nature, which we call them as a neoblast. And this neoblast, which are pluripotent in nature, gives the planaria the capacity to regenerate. So what happens is this: you take planaria, doesn't matter where you cut. For instance, in this particular case, if you cut the head of the planaria and the tail of the planaria, you just have a trunk region, and the stem cells in that trunk region identifies the anterior, identifies the posterior part. They starts differentiating into the progenitors. The progenitors first differentiate into tissues organize and regrow into a functional animal. So, and people have done, our Alejandro lab and Peter Regan lab and Rajeski's lab have done this extensive single cell analysis, and they have found that there is a pluripotent stem cell, and the pluripotent stem cells can differentiate into a committed stem cell, and each committed stem cell can give rise to a specific lineage or a cell type, and thus helps in the regeneration. So planaria gives us a beautiful opportunity to ask the question how stem cell state transitions are happening in the context of injury, and how these molecules are regulating the process of stem state transitions and also subsequently the process of regeneration. So what we have asked is three different questions here. We asked, are these tRNAs present in planaria? That's one question. If they are present in planaria, are they present in the stem cells? If they are present in the stem cells, how their biogenesis is regulated, right? So the three questions we asked. In order to ask the first question, are tRNAs present in planaria? What we did is we took the tRNAs and we took the planaria, we dissected them into thin fragments, which we call it as salami sections, from the head to the tail. And from each fragment, we have done small RNA sequencing to ask the question, how is tRNAs distributed throughout the animal? The second question is most obvious question. We took the planaria, we cut it into two halves, we took the regenerating tissue and asked the question, are tRNAs expressed there? But when we started addressing that question, we realized that the tRNA annotation in planaria is really bad. The first thing we have done is we have extensively annotated tRNA, and we have found that there are 457 tRNA species in planaria, and this is the distribution of anticodon arms on the tRNA, and we have constructed a codon table for tRNAs in planaria. We have validated these tRNAs extensively by Northerns in collaboration with Shiva from NCBS, and now we have 457 tRNA species. So we took these small RNAs and we mapped them to the 457 tRNA species and asked the question, how is the distribution of tRNAs across the tRNA population? And what we found is this: we found that a small percentage of tRNAs, which is around 1 to 3%, actually map to the tRNAs, right? And they are distributed throughout the animal rather than present in a specific region of the animal. And very interestingly, what we found is this: we found that there are two different species of tRNAs. One species which is 18 to 24 nucleotide, and we found another species which is 25 to 35 nucleotide. Now we ask the question, where are they coming from tRNA? We found that the 25 to 30 nucleotide is coming from the 5' arm of the tRNA. And if you look at 18 to 24 nucleotide species, we found that one species of 18 to 24 nucleotide is coming from 5' arm. And very interestingly, the second part of the tRNA, which is 18 to 24 nucleotide, is coming from the intermediate regions of the tRNA. That intermediate region starts from just after the anticodon arm and ends before the T-C loop. So we call them as intermediary turfs. So now this is the nomenclature. The 25 to 30 nucleotide species is called as 5' tRNAs. The 18 to 24 nucleotide species coming from the 5' arm is called as TUFs 5s, and the intermediary species are called as ITUFs. So that's a nomenclature. And what we found from this heat map here is they are distributed throughout the animal. They are not present in one aspect of the animal, right? So the next question we asked is, do these different tRNAs has a base residue preferences? We haven't found a specific base residue preferences for the 5' tRNAs, 18 to 24 and 25 to 35 nucleotide, because their start region is always the 5' end. The 5' end is highly enriched with G, so you see a very strong G preference. What is interesting with ITUFs is this. In case of ITUFs, we found that ITUFs have a strong preference for U, suggesting that the ITUFs might be processed by either dicer or organate species. So now we went back and asked the question, how are they regulated? All these three different tRNA species, how are they regulated during the process of regeneration? So, what we have done is we have taken the regeneration tissue at 3 hour, 12 hour, 24, 2 day, 3 day, 5 day, and 7 day. We did small RNA sequencing. One is we call head, that is regenerating the tail, and the tail that is regenerating the head. And we asked how is the distribution of 18 to 25 to 34 species and 18 to 24 species. And what we found is the one species that is heavily regulated during the regeneration is your 5' tRNA, which is 25 to 35 nucleotide species, rather than the 18 to 24 nucleotide species, suggesting that this 25 to 35 nucleotide species might have a potential role in regulating both the tail regenerating head and the head regenerating the tail here. So this is the 25 nucleotide species. And one thing that really caught our attention is the tRNA species that are heavily upregulated at 3 day and 5 day and 7 day post regeneration, suggesting that this tRNA species that you see that are upregulated at later half of the regeneration, that is especially the head that are the tail that are regenerating the head, suggesting that they might be essential for the process of neurogenesis. And we also found similar things for the TUFs that are 5' and also for the ITUFs. This is a correlative data which is suggesting that this tRNA species, all the three species, are dynamically changing as the animal is regenerating, suggesting that they might have a functional role in terms of regulating how the regeneration is happening. So currently we are doing antisense treatments on the regenerating animal to see how they affect the process of regeneration. But what caught our attention is the biogenesis of tRNA fragments. So why are we interested in biogenesis? If you take planaria and look for endoribonuclease, which is angiogenin that cuts tRNA in mammalian species, we haven't found angiogenin in planaria. So that brings us to the question, what is this endoribonuclease that is actually generating this 25 to 30 nucleotide species, which in mammals we know is the angiogenin, right? So one endoribonuclease that can potentially generate 25 to 30 nucleotide species in planaria is a PV protein. PVs are organate family of proteins, and my lab has previously shown that these PV proteins are highly enriched in the stem cell compartments. And we have also shown that if you knock down PV, you actually block the process of regeneration, right? So our guess is the PV proteins in planaria might potentially play a role in generating the 5' tRNAs. So how do we address that question? When we are trying to do this, there was a paper that was published from Kim et al. In Genes and Development, where he has taken the PV proteins from planaria, he has pulled down and asked the question, what are the interacting RNAs? And interestingly, if you look at this annotation, there's a huge chunk which comes as no annotations there. What this paper has shown is these piRNAs that are interacting with PV proteins are coming from the transposons, but they haven't really went back and looked at what these no annotations are. So now it's pretty simple. We went back and asked the question, what is this no annotations? Because we have an annotated tRNA database with us. And what we interestingly found is this. We found that the PV proteins in planaria indeed interact with the tsRNAs, suggesting that the endoribonuclease that actually generates the 5' tsRNAs in this particular case is none other than a PV protein. So this brings back to the question, is this specific to planaria? The answer is no. In mammals, I previously said that there are two pools of tsRNAs. One is angiogenin independent pool and other is angiogenin dependent pool. So now the question is, are PV proteins expressed in the stem cells of mammals? The answer is yes. There is a PV protein which is called PVL4, which is expressed in the stem cell populations, and they are enriched as the stem cells differentiate. So now we think the angiogenin independent endoribonuclease that cuts the tRNA to generate tsRNAs is none other than the PV protein, and we are currently knocking it down to ask the question, how does it change the dynamics of tsRNAs and thus change the embryonic stem cell state transitions. So this is one typical case where we used planaria to identify an endoribonuclease, and now we are using that endoribonuclease in order to drive the stem cell function in mammals. Now the other question is, we also have these two different fragments of tsRNAs. One is I-taus, which are 18 to 24 nucleotide species, and another is 5' taus, right? And if you look at I-taus, as I said, I-taus have a heavy enrichment for U at the 5' end, suggesting that the I-taus might be generated either by dicer cutting or by organate cutting, because both these proteins can cut and generate 18 to 24 nucleotide species, right? So we went back and asked the question, do we have dicers and organates in planaria? Yes, we do have dicers in planaria, and we have two organates in planaria. The EGO2, which you see on my screen, has been well characterized. If you knock down dicer and organate 2 in planaria, it leads to drastic regeneration defects. And we have also shown that both dicer and organate 2 control the microRNA pathway. And we have a second organate here, which is EGO1. So we don't know the function of EGO1. And EGO1 is interestingly expressed in the stem cell compartment and is also expressed in the other tissue types. When we knock down EGO1, we have seen a regeneration defect, but these regeneration defects are very mild, not as extensive as you see with the EGO2. So now we asked, if we knock down EGO1, do we see a defect in the microRNA pathway and a piRNA pathway? The answer is no. The piRNAs and microRNAs are unperturbed when you knock EGO1. So next question we asked is, does this perturb the tsRNAs or I-taus? What we found is, when you actually knock down EGO1, you see, sorry for that. When you knock down EGO1, you see a dramatic decrease in the I-taus levels, suggesting that the I-taus are actually generated by an endoribonuclease, which is EGO1 in planarians, right? So this is where we stand. We stand here. In planaria, the tRNAs potentially are processed by PV proteins, and we know that the PV proteins are enriched in the stem cell compartments. So the PV protein cuts the tRNA and generates 5' tsRNAs, and we think these 5' tsRNAs indeed go back and control how the neoblast population undergoes differentiation. In other words, it controls the pluripotent stem cells of the planaria, because we know PV proteins are enriched in that. The second one is this I-taus, which is processed by EGO1, generating I-taus. And we know that I-taus are expressed both in the progenitor populations as well as in the differentiated cells. And we also know that EGO1 is expressed in the progenitors and differentiated cells, suggesting that the EGO1 cleaved I-taus might potentially regulate the differentiation of progenitors into a terminally differentiated cells. So now you have this interesting phenomena where the tRNA is being broken down into two different species, And these two different species are regulating different aspects of the stem cell function. So this is where we started my talk, right? You have a stem cells that undergo transitions and differentiate. So what we have shown is you have the tRNA that can be split into multiple species, and we know that these multiple species can actually regulate translation. And by regulating translation, they're actually regulating how stem cells undergo transitions by differentiating into progenitors and further differentiating into a specific cell types. So that is where we are in the context of tRNAs and tsRNAs, and our understanding is still at the tip of an iceberg. We still don't know the details of how regulation is happening, how modifications are regulated, how the tRNAs are, how different parent tRNAs are being processed into tsRNAs, and that is where we are heading towards. But we are seeing all this in the context of how stem cells function is being regulated. So this is the acknowledgement slide. And the initial part of my work, which is ESC's identifying IGF2BP1, was done in collaboration with Dr. Shrikala Raghavan from InStem and also with Dr. Ramanuj Dasgupta from A-STAR. And it's a beautiful collaboration. We have published a couple of papers from there. Most of the tsRNA work done by actually two students, now both are postdocs in my lab. One is Shrikar, who has done an amazing work in understanding the function of this tsRNA. And literally every aspect of bioinformatics was done by Vairavan, who is another postdoc. He's standing right next to me there. And both of them has actually driven the whole tRNA project or tsRNA project in my lab. And now we can see how this whole project is evolving. And I also have multiple collaborations at InStem and outside InStem. I want to thank the funding agencies with this Swanajayanti intramural funding, Welcome DBT, and also DBT, and thanks to the NGS facility and the CIF facility. And I'll be very happy to take questions. Thank you very much.
Thank you. Thanks so much, Das. Really fascinating talk. We had some questions coming in the first 10 minutes, but then your further slides resolved those. So we'll go with some of the questions that have remained. Maybe it's best if people can ask their own questions. So can we unmute Deepakash? He has a question. Deepakash, if you're here, you can ask your question.
Surbi, Deepakash has left, I guess. So
okay,
we can move ahead.
It's still an question.
So I can see that question there.
Yeah, it's right there.
Yeah.
Ischemic mRNA is translated by cap dependent and independent initiation. IGF2BP1 is required in which case for c-MYC mRNA translation? I really don't know the answer. It's a very good question. I mean, c-MYC is a very interesting molecule where both cap dependent and independent functions are both cap dependent mechanism and independent mechanism exist based on the cell cycle in which it is. Our guess, based on what we are seeing, the tsRNAs might potentially regulate the cap independent processes. That's what we think. But we don't have a definitive evidences for that, and we are still working on it. Okay, so I mean, that's what I can answer for Deepakash question, right?
Yeah.
And should I go through the other two questions there?
Yeah, I think we have Rakesh's question right there.
Yeah, how tsRNAs distinguish stemness genes and spare a differentiation gene translation? That's a very good question, and that's where I say this is being regulated by its association with the RNA binding proteins. We think the RNA binding proteins that are interacting with the mRNA ribosomes and the tsRNAs are making that call, right? So that is our guess. We cannot completely rule out the association of secondary structures with the mRNA, right? So we are still working on it, and we don't have a definitive evidences for that. But based on what we have seen, the specificity is coming by its association with a set of RNA binding proteins, right? So I think this is the same thing that got pasted, and there's one more question I see here. Will tRNA antisense oligos used in some of the experiments also bind to the parent tRNA? And this is a very reviewer question that we also got. And we have shown that this tRNAs, this antisense oligos, really doesn't perturb the tRNA here. One reason why we think is the tRNAs are heavily folded molecules, and spatially they don't allow the antisense oligos to interact with them. That gives it's an edge to interact with the tRNAs. So that's what our explanation for that is, right? So, and there's a question from Kaushik. When using antisense oligos against tRNAs, what about the levels of whole tRNA? Have you seen any change in the whole tRNA level? We haven't seen any change in the whole tRNA levels at all. In fact, a very small proportion of the tRNA is processed into a tRNA. That is where it becomes interesting. How is that stoichiometry being maintained, right? How does a cell know what proportion has to be converted into a tRNA? So that is what we are trying to understand, and answer is no. We don't see a dramatic change in the tRNA levels. What will be the anonymous attendee? What will be the future approach and some good uses of this research? So
Applications.
application, right? So what we are saying here is tRNAs are essential for stem cell function. And we all know what will be the use of a stem cell, right? So there could be a potential where you can use tRNAs and drive stem cells to undergo differentiation. That's one aspect of it. The other aspect is the tRNAs are heavily enriched in cancers. And what we see is these tRNAs are also being now used as a biomarkers to understand what kind of cancers they are. In fact, we are collaborating with Ramanuj from A-Star to understand these processes. That's one aspect as a biomarker. And the other aspect is if tRNAs are going up in cancers, is there any ways to regulate tRNAs which actually goes back and regulates the cancer proliferation? So the other application of it is in the cancer biology, and we are still working on it. So, and there are a lot of other groups that are also working parallelly to understand how tRNAs regulate cancer biogenesis. So these are the two areas that I could clearly see. The third one is this, right? We also see tRNAs going up as the neural progenitors differentiate into neurons. And we have a very preliminary data to show that there are tRNAs also in the synaptic junction, suggesting that the translation regulation at the synaptic junctions can also be controlled by tRNAs, suggesting that that's another application I could see. And most of the mutations in tRNAs, which is leading to a changes in modifications, are metabolic disorders. So a lot of metabolic disorders are happening because of the mutations in the residues where it can potentially be modified. Now we can suggest that this is the mutations that are regulating the modifications must also be regulating how tRNAs are being processed, suggesting that the metabolic disorders might have a direct connection with the levels of how tRNAs are being made. We also think, and it has been shown by Sunil from InStem, that tRNAs can actually be a sensors for changes in the metabolites. And one aspect of it is change in metabolites might actually take the tRNA, break it down into tRNA. And thus tRNAs goes and functions in terms of how state transitions are happening. So that could be another application that I see, right? So these are some of the applications I could see in the context of how tRNA field can evolve. So there is another question from Sudipta, a small query. When the fragments are made, can they rehybridize themselves to fully functional tRNA? Answer is no, because they are fragments. How even a nick can tear apart a hydrogen bond in the tRNA, and if it requires additional factors, my guess is you require additional factors, such as proteins that bind to the tRNA and separate them off. So that's the answer for that, right? Jyotin Gogri, Apart from tRNA fragments, which have functioned during differentiation, rest of the portion of tRNA are degraded. Do you think there is a preferential protection of functional turf from degradation? Yes, the tRNA ends can be modified, and that modifications can prevent the endonuclease degradation, right? So that's the answer for that. Again, there's a question from Sudipta. What proportion of tRNAs will go for this non-canonical function? I mean, we haven't really calculated the proportion. The rough estimate we have is 1 to 2%. So that's the answer. Nice talk. Thanks for that. Is 5' tRNAs is regulating c-MYC mRNA translation by a structural mimicry or allosteric writing? This is a beautiful question. I don't know the answer. We are working with Vinod, who is a cryo-EM person, to look at the structural association of tRNAs with the RNA binding proteins and the ribosomes. Hopefully, we'll have the answer. What pool of antisense tRNAs are chosen for the study? The one that we choose is, we have chosen the top 5 enriched tRNAs. I mean, I have the names there. One of them is glycine. My understanding is that, right? The tRNAs that are derived from the parent glycine chain. So, so that's it. I think I answered the questions.
I think
so
we had a hand up. Pradeep, you had a question?
Yeah, actually, he answered my question.
Okay, we also have some questions on YouTube. So, did you observe any epitranscriptional modifications on the tRNAs?
Epitranscriptional modifications on tRNAs. I mean, tRNAs are modified. I'm sorry. I mean, this is you're saying that does tRNAs are contributing epigenetic changes?
I think they're looking for epigenetic angle. That would be.
Yeah, well, because of course, I mean, tRNAs are modified. That's what I was talking, right? Yeah. So if there are modifications on tRNA and the tRNA is being broken apart, that tRNAs might be carrying a modification. That's one part. And as I said, angiogenin, when it cleaves, it cyclizes 3' and 2' and 3'. So that's another aspect to it, right? Yes, if the question is about epigenetic regulation, tRNAs have been shown to contribute in epigenetic regulation, right? There are these 2 science papers that were published, which shows that there could be a potential role for transcriptional regulation of this tRNAs, right? So a lot of work is happening in that direction.
Yes, that would be interesting to follow up.
Yeah. In fact, I mean, I haven't said that. I mean, we are still looking at it. If you pull down 5' tRNAs, one of them is ribosomal proteins, and other protein that we also see enrichment is histones.
Yeah.
So I was talking to Rakesh about it, saying that maybe one way tRNAs function is by just quenching histones, leading to opening of the genome, right? So, and tRNAs are also heavily upregulated during the early embryonic development, where a lot of opening and closing of the genome happens. So that's another area that I'm very much interested in. Yeah. So go ahead.
Yeah, I think Rakesh has a hand up.
I'm not able to figure out whether I can. Can you hear me?
Yes, go ahead, Rakesh.
Yeah.
Okay. This is a new kind of Zoom. I'm not finding the buttons here. Anyway, so I wanted to ask. That image was wonderful, and it's really crazy the way things are progressing. RNA doing all sorts of things. So I just wanted to know whether there's evidence that these RNA, once they are made, they are fully processed and they are employed for, deployed to do translation. And then they are then broken into 2-3 pieces for the regulatory role. Or there is a different category when they are made, they are straight away processed for regulation. And during, if they are going through translational route, they get epigenetic, or covalent modifications on them, which makes them more special and more specific or something. There's lots of amazing things that come to mind, right? So, what do you think of it?
That's all are beautiful questions. Unfortunately, I don't have many answers to that. That's where I said we are really at the tip of the iceberg in terms of understanding the biology behind this tRNAs, right? But what we guess is the tRNAs are being matured when they are being processed. That's what I guess, because there was also studies that were published by different groups where they said that if you change the urinylation at a 13th residue on a tRNA, you can actually change how the tRNA is being processed into an 18 to 24 arm versus a 25 to 35 arm. It's a beautiful paper that got published, suggesting that the modifications play a critical role in deciding how the tRNAs are being processed. So if the modifications are deciding how tRNAs are being processed, I believe that the mature tRNA arm is the one that is actually being processed. So, but we don't have any concrete evidence to show that is how it is. And angiogenin, in spite of being in the nucleus, the processing we think is all happening in the cytoplasm, suggesting that if the tRNA is coming out into cytoplasm, that could be a mature tRNA which is actually being processed by angiogenin. So, but these are all speculations, Rakesh. So I don't have a definitive answers for that. But these are the directions which tells us a lot about the biology of both tRNA as well as the tsRNA. So this is what I can explain now. Yeah.
Thanks.
Thank you, Rakesh.
Santosh, you had a comment?
Yeah, I just wanted to ask, what is happening to the remaining half of the tRNA? We are talking only about the 5' case.
Yeah. So in a sequencing, we haven't caught the 3' halves. When we do the 5' halves, so there are two things to it. That also brings, interestingly, Rakesh's question. We haven't really tested it. If the tRNA is activated, that means the amino acid is sitting, we might have missed the 3' half because there's an amino acid on it and we were not able to sequence, right? But then have you found any species that are smaller species coming from the 3' half? We haven't found any. So for now, we believe that the 3' half is being degraded. Yeah, so there are protocols to remove amino acid and sequence them, right? And I think we will do that subsequently.
Yeah. One more question I have is just like, once the tRNA is amino acylated, how it just is pulled by EFTA, elongation factor, and how these tRNAs are rerouted from translation for the kind of function.
Yeah. See, I mean, this is where I say, right? I mean, if you have 1,000 tRNA molecules of a specific parent tRNA, right? The question is, all these 1,000 molecules are modified at the same rate? My guess is no to that. So the ones that have differentially modified based on the availability of a metabolite is a deciding factor how these are being processed. In a ways, this is again, I'm just speculating here. In a ways, the concentration of the tRNA acts as a sensor for the availability of a metabolite, right? So it's a game of, it's all stoichiometry, right? I mean, what is the proportion of a specific tRNA that is present and how much of the tRNA is being modified is determined by the availability of a metabolite and an enzyme. That decides how much is the tsRNA is being processed. And that tsRNA is interestingly going back and regulating translation, because we don't see a massive depletion in the tRNA population from which the tsRNAs are being made, right? Suggesting that it's a proportion that is being made, right? And yes, I mean, the answer is we're still trying to get a hang of how these proportions are determined, right? That's what the whole research is about. Yeah. Thank you. Yeah.
I think I don't see any other questions. Just a future direction, thus, you mentioned something with the PV link, right? That PVs that you got from the planaria. So you're planning to look into this further with knockdown?
Yes, we are knocking down PVL4 from ESCs and ask the question how they regulate ESC transition and how it affects tsRNAs. But the bigger question is this, Rabi, like you have two endoribonucleases. We know both are in stem cells now, right? And both could potentially generate two species of tRNA. Now, the question comes is, how is that regulation happening? That is what is mind-boggling to me, and we are trying to look at it.
Yes. All right. Wonderful. Okay. I don't think there are any other questions. Thank you so much, Das. This has been a great session.
Thank you very much. I really enjoyed talking and interacting. Thanks a lot for inviting.
Pleasure's us. Thank you so much. Thanks everyone for attending. See you next dialogue. Bye-bye.
Bye.